Okay, thank you so much. Thank you, the organizers, for inviting me to this talk. I'm very sorry that I couldn't come because of my heavy teaching load. So it's certainly my honor to be here in the IBS, maybe in the future. So for the sake of time, I'll just directly go to my talk. As my topic has mentioned, I'm talking about topological defects in active liquid crystals. So the outline of my talk will be the first I will introduce what is topological defects and what is active liquid crystals. And then I will talk about the new features that we can see from all these systems. And in the end of the talk, I will talk about some ongoing efforts that my group is working on. So nowadays, liquid crystals becoming increasingly more interesting to a general audience. Because in the past 5 years, a lot of liquid crystal phases have been observed in non-traditional systems. Here I gave you two examples, two biological systems. One is epithelial cells, the other is progenitor cells. So in all these two examples, you can see that the cells will elongate into anisotropic shape, so they acquire a orientation, and the nearby cells, they tend to align with each other. So this is the typical feature of liquid crystals. And occasionally, you're going to see that regions that are disordered in its orientations, which are schematically plotted in terms of these two pictures. We call it a +1/2 defect and a -1/2 defect, which I will be more specific. So what happens in all these biological systems is that they can see that the cells align in certain directions, and they can also see the spontaneous motion of those topological defects. And on top of that, they can also see that cells can also pretend to die at the defect cause. And in the right picture, they see that the defect near the defects, the cell density will be either very high or very low, according to the feature of the defects. So it seems that topological defects have some biological consequence. And in the theory, we have already predicted that in the active systems, all these systems that are active because the individual constituents can move or divide by themselves, on their own. So these defects can be mobile in these systems. And in the more traditional liquid crystal research, there's a new trend in using liquid crystal defects to organize particles and molecules. So here I give you two examples. In one example, topological defects are used to direct self-assembly of colloidal particles. On the right, topological defects are used to template the assembly of amphiphilic molecules, so that eventually you can see that we can obtain a freestanding polymer ring after the raise of the temperature, so that the molecules can leave the topological defects and form a certain shape. So these recent advances in liquid crystal show the importance of topological defects in guiding the self-assembly of particles and colloids. We want to move forward to ask a more advanced question. Can we use topological defects to control a transport phenomenon in microscopic systems? So here I give you a schematic of actually an animation of the inner cell, in which that we can see that the motor proteins are carrying cargoes and working on top of a microtubule. So this is a very rigid and long polymer. So this is the system that we recent researchers are working on, especially Professor Zhang from UCSB. So they use this type of systems. They extract the microtubules and the motor proteins. Here they call it kinesin motors. So the kinesin motors can cross-link two microtubules, and the two microtubules can actually slide on each other due to the action of the motor proteins activated by the ATP hydrolysis. So that will, if you put that system on a liquid interface, more specifically an oil-water interface, actually the microtubules, because they are very rigid and very long polymers, they form a liquid crystal phase. And because of the action of the motor proteins, these biopolymers actually constantly move around. And you can see a spontaneous flow formation and also the motion of defects. And occasionally you can see nucleation of a defect pair as indicated by the two arrows. We actually worked with another group at the University of Chicago, Margaret Gardell, by using another type of biopolymer called actin filaments. And the corresponding motor proteins are called myosin motors. And we also see a very similar phenomenon that we can observe spontaneous flows and the self-propulsion of topological defects. So these are the two experimental systems that our theoretical and computational work relies on. So as we collaborate with Margaret Gardell group, things that we have confirmed from the experiment is that in the simulation of theory, we can see that topological defects may adopt different shapes according to the material properties of the liquid crystal. And indeed, in the experiment, very simply, because this is a biopolymer, so they can easily change the average length of the filament. And by doing that, they can also systematically see different shapes. So they can either see a U shape or a V shape according to the material properties. Actually, more interesting is that if we add, our experimentalists add motor proteins to the system to activate the liquid crystal, they can see that the shape of the defect has changed from a V letter to a U letter, indicating that the bending mode is softened during the excitation of the system by the action of the motor proteins. So in the active systems, the apparent material properties will be different from the passive properties, and this is reflected by the morphology of the defects. So I want to add one more sentence to this picture, is because that in many traditional systems, to measure the mechanical properties or mechanical constants of a system, you have to add external strain or external field to measure the response of the system to extract the mechanical properties. But the nice thing about the liquid crystal is that you can just inspect either the shape or the statistics of your topological defects, and you can also extract useful mechanical properties. In this case, the ratio of the bend constant and the spay constant. So this is a very convenient way to infer the mechanical properties of a elastic system. So in order to understand the flow and the motion of the defects, we have to consider the Navier-Stokes equation. On top of that, we have to also describe the evolution of the microstructure of the liquid crystals. So here is our simulation framework. So we have two fields. One is the direct field, which is actually we solve using the Q tensor. The Q tensor gives you more information about the liquid crystal. And we have another equation, which I call it a momentum equation, in which we solve the evolution of the flow field. And the important feature of these two coupled equations is that in the stress term, in addition to the passive stress, which accounts for the anisotropy of the material, because the liquid crystal is an anisotropic liquid. On top of that, we have an active stress term. So this active stress term turns out to be proportional to the nematic order parameter Q tensor. And this alpha tells me how much my system is away from equilibrium. So we can treat alpha as the amount of motor proteins in the system. So without alpha, the system is like a passive system that every external stir will eventually die out. The system will dissipate energy and eventually reach some equilibrium. But with the active stress term, and then the system can be sustained in terms of the motion. And we actually, the first example that we look at is a spherical system. On top of it, you can see there are 4 defects. So this is due to topological reason. So the ground state of a liquid crystal confined on a sphere is composed of 4 defects. And on this sphere, the microtubules also move around continuously. And that gives rise to the collective motion of the defects. it turns out that these defects oscillate periodically. They oscillate actually between two modes. One is called a tetrahedral mode, that the 4 defects organize into a tetrahedron. The other is the planar mode. The 4 defects actually reside within a plane. And by measuring the average angular distance alpha, which is defined as the average of the 6 angles alpha ij, which is the angle between the 2 radii connecting the 2 defects with the center of the sphere. So we can calculate for the 2 modes, these average angular distance will be different. And experimentally, the measurement shows that the system periodically oscillates between these 2 modes. And the periodicity is around 100 seconds. And in the original science paper, they have introduced a very simple model. They treat these defects as point particles interacting with each other through a repulsive force, and they can reproduce the experimental results. So our job is to use the more sophisticated continue the hydrodynamic simulation and to redo the calculation. And it turns out that the trajectories are different from the original paper. So on the left, that's the theoretical prediction of the original paper, that 4 defects form 2 closed loops. And our trajectories are more complicated. The defects actually working on a deformed cube on the sphere. And there is a follow-up work, which is a slightly different experiment that we still have these 4 defects working on the sphere, but outside of the droplet, we have another liquid crystal. And this liquid crystal can generalize a defect ring. And this defect ring is dancing back and forth due to the hydrodynamic flow that is generated by the defects. So in this work, they have used our trajectories and they can reproduce quantitatively the dancing mode of the experiment. So our model is now benchmarked by this very nice experiment, and also the our predicted trajectories can well explain a new experiment. And so the fundamental difference between our model and the original model is that we have included the hydrodynamic interactions between the defects. So with that, we want to go further because this is a very simple system. It's a very symmetric system. So actually, in the next, we show you a more non-trivial system. In this work, we collaborate with Francesco Sages and Teresa Lopez-Leon in Europe. And so they can use different droplets. Actually, here they use a spheroid droplet formed by HCl molecules. So outside the droplet, they still have the aqueous solution of the microtubules. So you can see the texture of the microtubules. And these microtubules can also condensate on the interface of this droplet. And they can also form 4 defects. And what's interesting is that these defects also move collectively on the spheroid. And it turns out that our model can reproduce the trajectories of the experiments. So as you can see that the 4 defects form 2 pairs. These 2 pairs are nearby the 2 poles. So they spiraling near the poles, and occasionally they stop spiraling and then they move longitudinally to exchange positions. And if you focus on the North Pole, and you can see that each time when the 2 pairs are close to the poles, the defect pair is actually rotating clockwise. Okay, actually, after a while, when they exchange position, the new pairs are also rotating clockwise. And qualitatively, we can see that also in the simulation, that the 2 defect pairs initially spiraling near the poles and then exchange positions. So these trajectories are very similar to the trajectories that we have obtained from a spherical system. But there's a difference because in our simulation, the handedness of the defect pairs will change alternatively as they exchange positions. So these 2 pairs, they rotate clockwise, but when they exchange positions, and they actually rotate counterclockwise. So to account for the difference, we introduced an isotropic friction because this is an adhesive droplet in the symmetric phase, and we have the symmetric layers that provide anisotropic friction to the hydrodynamic flow. By including that anisotropic friction, and then we can capture this physics that the hardness of the defect rotation will be preserved in the simulation. So we are very happy about this result. And actually, there's an additional physics that we see from this system, which is that there's a rigid body rotation. So as the defect pairs are rotating, actually they are rotating in different hardness, and this can create a hydrodynamic flow. And when they are very close to a substrate, this hydrodynamic coupling with the surface can induce a rigid body rotation. And so this is very similar to the dunkings that grinding the widths. Okay, very similar. So the mill is your droplet, and the width will be your surface. Okay, so this is not this actually have already been discussed in other systems, especially bacterial. So when the bacteria is very close to a substrate, they also exhibit circular motion. And indeed we have evidences from the experiment. So as you can see, if you look at these two droplets, the defects of their poles are rotating with different hardness, and the hardness of their rigid body rotations are also opposite. And also another thing is that when the defects are moving longitudinally, you can see that the rotation, the rigid body rotation will cease. And once the defect is spiraling, the rigid body rotation will continue. So these are all consistent with our theoretical prediction. And in addition to that, we can also see that sometimes there are multiple defects on the droplet. And if that is the case, there will be no persistent rigid body rotation. So this is again consistent with our prediction. So this means that we can have a very rich defect dynamics on curved surfaces. And we want to have a more flexible control parameter to manipulate the defects and flows, because for this confined system, defects have nowhere to go. So they have to be on the surface. So we want to study a flat interface. So to do that, actually our idea is to introduce activity patterning. So in the simulation, we create a system. On the left, the active stress is very low. On the right, active stress is very high. And in this case, we can generate a lot of defects on the right-hand side. And it turns out that those defects tend to stay in the active region. So it seems that this activity boundary, this is not a real boundary, but the activity boundary seems to confine our defects. And very interestingly, for the sake of time, I'll skip that. And experimentally, we do observe the phenomenon that the experimentalists, Margaret Gardner and Zach Bryan from Stanford University, have shined light on this red box, and we can create a lot of flows inside the red box because this region is activated, and the defects seems to be confined within this region. Okay, actually we can go further in the simulation. So simulation predicts that we can use a triangular activity pattern to create a defect pair. This is very nice because we can use that to plant defects on the system. And another thing, which is very similar to what I just mentioned, is that we can use activity pattern to guide defects. So on the left is a uniform active system that the defects will move horizontally. On the right, we can see that if we only activate the red box, the defects are actually deflected by the box and move upward. And this is actually confirmed in our experiment. So here you can see that we use a quarter annulus. And for each of the 5 independent experiments, our collaborator have seen the guidance of the defects by the activity pattern. So this confirms our simulation predictions. So actually, we want to go further. We consider a microfluidic device, and we can actually change the destiny of the defects. So we first use a triangular pattern to create a defect pair. Then we use different activity pattern to guide the defects into different branches of the system. So you can see this. So eventually, one defect is in the top channel, the other one is in the bottom channel. And actually, we can explain that by looking at the local active force by taking the divergence of the active stress. Actually, the gradient of the pneumatic order can create active force. In addition to that, the activity gradient can also induce an active force, and this active force is actually is on the activity boundary. So, which is labeled by the green arrows. So the green arrows will actually repel the defects. That's why the defects are repelled by the activity boundary. And on the right, I show you the triangular pattern. And this active force also is consistent with the direction of the defect nucleation. Okay, so in the next few minutes, I'll directly go to our ongoing work for the sake of time. So now we are interested in the active defects, and actually we confine the defects on a cylindrical system, and you can see that the defects is spiraling. So the red is a +1/2 defect, the green is a -1/2 defect. So this is something that we are working on. There's another thing that we consider is that we are not only interested in guiding defects, we're also trying to deform defects. So this is the experiment. We use optical trigger to change the orientation of the anchoring pattern on the surface, and with that we can deform our defects and reconfigure our defects. So, and this is already published, but we are still working on that to have a more flexible systems. So. So with that, I want to conclude. We have built a active crystal model and also experiment to study the defect dynamics in the confined system and also in the system with activity patterning. And we see that there's a new physics that comes from the activity gradient term. And lastly, something that I didn't show is that we can use our control to perform logical operations over our defects, so that we can treat the defects as electrons, so that we can transmit information and also to perform certain operations using materials. So I want to acknowledge my Chicago collaborators and Professor Juan Di Pablo, and also my students are also contributing to all these slides that I have shown. So thank you very much.
Thank you for the nice talk. Do you have any question? Yeah.
Okay. Haru, very good talk. I have a question. When you mentioned that the simplified description of this experiment for defects on the sphere does not agree with your full simulations.
Yeah.
What I'm wondering, how can you improve this simplified description? In this work, the description was oversimplified. I agree, but you probably can improve it and add something to make it more consistent. I think you don't need to solve the entire system of equations to reproduce this motion.
Yeah, I think Thank you for the question. Yeah, I agree that maybe we can have a more sophisticated interaction potential between the defects, and with that we can have a better description of the defects. Because as you can see that the trajectory is too simple. So in the original work, so and if we can account for more details of the interaction potential, maybe we can see a more chaotic, more random motion of the defects.
Yes, I would think so, yes. You need to include also the flow generated by the defects, then probably you can capture it.
Yeah, exactly. Yes. Yeah. Thank you, Professor Ernst.
And other question, yeah.
Hello, thank you for the nice talk. So maybe I missed. So when you mentioned about the controlled motion of these defects, so what is the role of temperature here? Is like
Oh, so this is a asymmetrical system because we're working with biopolymers. So there's no, we do not change temperature. It's in the room temperature.
okay. And do you
And
expect any changes with this when let's say a finite temperature or so?
Actually, so if we rely on the biopolymers or bacterial, so these biological systems are very sensitive to temperature. So if you change temperatures, maybe they can be frozen or they don't want to move. So, but if we look at more synthetic systems, and I agree with you that the temperature will play a role. Actually, I have a current work that we use a temperature gradient to guide defects. So we actually, our simulation predicts that the defects tend to move to a hotter region. So that maybe that's something that in the future we can use synthetic active crystal to explore.
Thank you. Yeah.
Thank you for your question.
Any more question? Yeah, I have a simple question to you. You talked about two kinds of defect, plus 1/2, minus 1/2. Is there any more different kind of defect? Is it possible or not?
Yes. So the defects that I'm talking about are 2D defects. So in 2D, the charge is usually half integers. So plus, minus 1/2, or plus or minus 1. So for example, plus 1 will be just a radial defect. So this for 2D system, you just use a half integer or integer. You can classify all the defects. And in 3D, the situation will be more complicated. So as I have shown you in the last system, you can see that these are the discrimination lines in 3D. So in this case, you can have very complicated defects, and the local feature of the defect can be very different. So you can have line, you can have loops, you can have a lot of other. You can even have plane defects.
The line has one invariant topological number.
Yeah, yes. And locally, you can also you can see that we use a angle beta to characterize the local winding on the line. So the winding can also vary along the line. So it's very complicated. It's much more richer than the 2D defects.
Yeah, thank you. I have one more simple question. Is it possible the two same defect, I mean plus 1/2, can meet or not?
Usually they are elastically repelling to each other. Yeah.
Okay, thank you.
So it's very difficult to put them together. Yeah.
Any more question? Okay, thanks, speaker again. Thank you very much.